Hello, my name is Jeff Millman. I'm an assistant professor of medicine and biomedical engineering at Washington University School of Medicine. Here, I lead a research team made up of bioengineers and biologists that are united in using stem cells for the study and treatment of diabetes. Diabetes is caused by the death or dysfunction of the insulin-producing beta cell, which is found within islets of Langerhans in the pancreas. Diabetes progression is caused by the loss of these healthy beta cells through their destruction, such as by autoimmune attack, or by their dysfunction, which can be caused by stress or genetic factors. Patients with diabetes are commonly treated with administration of exogenous insulin. And while this is indeed a life-saving therapy for many people worldwide, insulin injections and drug therapies do not actually cure the diabetes, but is instead an imperfect treatment. And these imperfections in the way that insulin is administered can lead to poor glucose control that causes long-term health complications. A potential long-term functional cure for diabetes is replacement of the endogenous diseased cells with exogenous islets containing these insulin-producing beta cells. When we look at the diabetic population worldwide, we see that there are hundreds of millions of people that suffer from this disease. And what this tells me as a cellular engineer is that we need a large-scale sustainable source of replacement cells and tissues to even begin to scratch the surface of the number of individuals that could benefit from a cell replacement therapy. So my team focuses on the use of stem cell technology as the source of these replacement cells and tissues. Stem cells have the ability to undergo a process known as self-renewal. That is, they can proliferate and maintain their stem cell state virtually indefinitely. Or if given a different set of factors, they can undergo a process known as differentiation to become any terminal differentiated cell type within the body. This includes the insulin-producing population. The way that we achieve this is to recapitulate embryonic development inside of the laboratory. My team uses a variety of cell culture formats in order to generate these cells, including bioreactors and high surface area multi-stacks of plates, to in the end produce a final product that consists of cellular aggregates that are about the same size as an islet, but also contain the insulin-producing cells. So on the screen is a schematic of our process for making these stem cell derived beta cells. We can first go and start off with the pluripotent stem cells that we want, and these can be generated from patients through reprogramming. After we have the stem cells, we can direct their differentiation to beta cells through a multi-stage differentiation protocol that's designed to mimic development, going through the various types of progenitors and intermediates that the cells would normally go through inside of the body, but instead doing it inside of the in the lab using the cell culture vessels that I mentioned. And this is achieved by giving them different combinations of small molecules and growth factors in order to be successful with this. So we've been doing this for many years, and we have to generate the refined process that I'm going to be talking about a little bit today. We've been most recently interested in going beyond targeting transduction pathways, which is very commonly done in the field, and trying to look a little bit more broadly into how the microenvironment could influence this choice to become a pancreatic cell. And this is work that was done recently by Nathaniel Hogerby and Pun Agtor-Warowat in my lab, and published in Nature Biotech earlier this year, if you're reading more on this. And what they had found is that this key stage here, stage 5, which takes proliferating pancreatic epithelial cells and converts them over into the actual endocrine cells or endocrine progenitor cells, we found a very important role of the actin cytoskeleton in this process. And this development of this step of the process came from this key insight that we found that with decreasing stiffness of the substrate the cells are attached to, that our differentiating cells are capable of having elevated levels of gene expression of key transcription factors that are responsible for differentiation to endocrine cells, including NEROG3. Based on this insight that synthesis was important here, we did a small molecule screen and identified the compound, the trunculin A, which is a actin deplumerizer, as a very potent endocrine inducer. Here's an image shown here showing a very low level of actin, which is the polymerized form of actin, present in cells after the trunculin A treatment. And if we look at the expression of these endocrine genes, again, we find a dose dependence of the expression of these genes with higher levels of the trunculin A treatment. Studying this new insight into the cytoskeleton a little bit more, we performed single-cell RNA sequencing, taking our progenitor cells and either not treating them with anything, treating them with trunculin A, again, to deplumerize the cytoskeleton, or treating it with nocladazole, which actually causes hyperpolymerization of the cytoskeleton, to see how this affects what cell types we produce with our process. If you're not familiar with these sorts of plots, these are TS&E plots where each dot represents the transcriptome of an individual cell, and dots that are more closely clustered together are cells that are more similar than other cells, dots that are further away. And based on this, we can identify what kinds of cells are being produced based on their gene expression signature and the clusterings that are occurring. With this particular analysis, we were able to identify a variety of progenitors and endocrine, exocrine cells, in part defined by their expression levels shown on with the violin plots down here, with genes characteristic of pancreatic progenitors, endocrine, and exocrine being observed for the corresponding cell populations. Now, looking at the contribution of these cell populations, depending on how we had treated the cells, we found that the vast majority of the untreated cells were identified as pancreatic progenitors here in blue. Cells treated with trunculin A were either early or late stage endocrine, as shown with the orange and the purple. And nocladazole actually went the opposite way. These were mostly cells that had an exocrine cell signature, shown here in red. And this is important because the exocrine and the endocrine compartments make up the entirety of the pancreas, and it's common actually to get exocrine cells as a unintentional off-target cell population in the process of making our endocrine cells and our beta cells. And so this led us to this understanding that you need essentially a Goldilocks region of actin polymerization if you want to have pancreatic progenitors that express key factors such as NKX61. If you hyperpolymerize the cytoskeleton, you'll actually go and make the wrong cell type, exocrine cells, which give rise to digestive cells. Or if you depolymerize it, you could actually make endocrine cells, and this is where the beta cell population is able to come from. So using this understanding that cytoskeleton is very important for making endocrine cells, we were able to then take these endocrine cells and further differentiate and mature them into stem cell-derived beta cells through a combination of permitting TGF-beta signaling, endocrine, using an enriched media that we had defined, and also resizing of the three-dimensional arrangement of the cells. So in the end, we have now a process that is able to make about 50% of the cells being beta cells. It's being defined as being a C-peptide or insulin-positive cell and co-expressing NKX61. Almost all the cells in the end of our manufacturing process are endocrine cells, as defined as being chromogranin A positive. And most importantly, these cells are functional. They're able to secrete elevated levels of insulin in response to a high glucose stimulation. And this is all done without sorting or selection. Not only are these cells able to respond to elevated levels of glucose by secreting insulin secretion, increasing their secretion of insulin, they are also able to display proper kinetics of the release of their insulin. So these are SE beta cells made from four different cell lines, one embryonic stem cell line and 3 iPS cell lines, where we perfuse low glucose solution over the cells to get a baseline level of insulin secretion. And then we switch perfusion from low to high. And what you would expect to see for a real beta cell is you would have a very fast response of insulin secretion, which is called a first phase response. And then we would have an elevated level of secretion as long as the cells are kept at high glucose, which is called a second phase response. And then returning it back from high to low glucose, you would expect that insulin secretion would return back down to a baseline level of secretion. And this is something, a very important functional feature of beta cells that have been missing in prior publications, but our cells are now able to do this. We had also recently found out that this functional maturation is being driven by the transcription factor 6-2, which is interesting because it's a transcription factor that is expressed by human beta cells, but is not expressed by mouse beta cells, which made it previously underappreciated in the field. We found that with time during stage 6, that we had a correlation of insulin secretion and the expression of 6-2 in our stem cell-derived beta cells. And we used CRISPR to create stem cell lines that were knocked out for the 6-2 gene. We found that we could still differentiate these cells and we could still make beta cells and they could still make insulin, but they had no ability to respond to glucose. So in the parafusion experiment, they had no response, no kinetic response to high glucose stimulation. And in the statics assay, they had no increase in their ability to increase insulin secretion. Most importantly, these cells are able to perform well in a preclinical model of diabetes. Here we took mice we made diabetic with STZ, which is a drug that kills the endogenous beta cells, transplanted them with our SE beta cells to see if the mice had reversal of their hyperglycemia. And then we were also able to remove the graft through a nephrectomy, which is where the cells are transplanted to the kidney, to ensure a return back to a diabetic state. This is showing our controls in this experiment that we induce diabetes, a very high level diabetes in these mice, and they don't spontaneously cure during the observation period. And we could transplant either SE beta cells made from an embryonic stem cell line in blue or an iPS cell line in purple, and we have a speed of cure that's comparable to an equivalent amount of human islets from deceased donors that were transplanted alongside of them. And again, removal of the graft results in a reversion back to a diabetic state in these mice. When we removed these kidneys, we're able to analyze them histologically and observed large regions of cells expressing C-peptide or insulin underneath the kidney capsule. Over here on the left is the kidney proper. You can see the kidney capsule there on the right. And our cells sit in between this region to be able to perform their physiological function. We also will see other islet hormones such as glucagon, which is red here, indicated as GCG, and also somatostatin shown here in red as SST. In addition to the hormones, we also see other key markers of beta cells such as NKX61 and PDX1, which are very important transcription factors. So even though we've had a lot of success recently in terms of making and maturing stem cell-derived beta cells, there are still some remaining challenges the field is facing. The cells we make are fairly mature in terms of being able to cure animals, but they are still immature when you compare them against actual primary adult beta cells. This is both in terms of their function and their transcriptome. It appears that the cells that we're making right now are more like juvenile or prepubescent cells. It is unclear right now how to actually mature them to be equal to an adult beta cell. Also, it's been difficult in the field to be able to transplant these cells in a way that could protect them from the immune system. The animal model that I used here was immune compromised. It didn't have much of an immune system, so it could tolerate transplantation of foreign cells. But of course, for clinical translation, there aren't many people without an immune system, and we want to avoid the use of immunosuppressant drugs. And so being able to transplant SE beta cells while allowing for them to perform their function and also protect them from the immune system at the same time is a remaining major challenge in the field. And then as work in this area continues, we're going to have to start thinking more seriously about clinical manufacturing of these cells, taking it from a research R&D process to an actual GMP and scalable process. And if you find these remaining challenges interesting, we have multiple openings currently in my lab at all levels. And so please send me an email if you are interested. So I really appreciate you listening to my virtual AIChE talk. The work that I showed today has been supported by the JDRF, the NIH, and through the Centene WashU Personalized Medicine Initiative. I'm very grateful for my wonderful team here at WashU who have been working tirelessly on this work. And if you have any questions, feel free to email me, tweet me, or I will be in the live chat. Thank you.